Welcome back to the deep dive. Today, we're embarking on a bit of an evolutionary journey. We're going to look into the deep history of modern drug targets.
That's right. Our mission today is really to understand the sort of ancestral toolkit of receptors. We're tracing the molecular roots of these crucial biological sensors way, way back through the tree of life.
And sometimes to some really startlingly simple origins, right?
Yeah.
I mean, like single-celled bacteria.
Exactly. We're looking at how nature repurposed these, very basic molecular building blocks into the sophisticated signaling machinery of the brain, the immune system, the whole endocrine network.
It's a remarkable stack of sources we're digging into today, and they really challenge some long-held assumptions in pharmacology and molecular biology.
They absolutely do. I think the core realization here is that our most essential receptor systems, they aren't, newly invented pieces of high-tech machinery. They are molecular mosaics. Chimeras, really. Cobbled together from modules that often evolved hundreds of millions of years ago for radically different jobs.
Okay, let's unpack that concept right away then. The idea of modularity. The sources really hammer this point home that many complex proteins in eukaryotes, so in complex life, are built this way. Why is recognizing this structure so vital?
Because it just simplifies the entire evolutionary pathway. I mean, think about it. Instead of needing to evolve a whole 100 kilodalton protein from scratch, evolution can just, swap in a pre-tested functional brick.
So it's like Legos.
It's exactly like Legos.
Yeah.
Modularity means large regions of the protein sequence are associated with specific functions.
Yeah.
You have a binding site here, a catalytic domain there. If evolution needs a new ligand sensor, it doesn't invent one from nothing. It just grabs an ancient, highly successful template and, modifies it a little.
We see this principle clearly in other areas, don't we? Like with voltage-gated ion channels, the ones that react to electrical potential.
Oh, absolutely. For those, we have these really clear lineage trees that connect, potassium channels, sodium channels, calcium channels. It's a neat family tree. But when we shift our focus to ligand-gated ion channels, or LGICs.
The ones that respond to neurotransmitters, GABA, glutamate.
Exactly. When we look at those, the history gets significantly fuzzier.
Fuzzier how? What do you mean?
Well, the LGICs, particularly in their modern form, they seem to be relatively recent eukaryotic inventions. We haven't been able to identify any clear, direct, prokaryotic precursors for the entire complex.
Which strengthens this idea that the sources are calling
the chimeric hypothesis. Exactly. It posits that these crucial signaling pathways, particularly in our brains, aren't monoliths. They're molecular mosaics, products of chimeric genes that were assembled through processes like exon shuffling.
So literally splicing together components from completely different evolutionary lineages.
That's the idea. And it totally sets the stage for discovering these really disparate, surprising origins for all the different parts of a single receptor.
And wow, nothing demonstrates that hypothesis better than the drastic revision needed for our understanding of the ionotropic glutamate receptors, the IGLRs. This is where the story gets really, really interesting.
It is arguably one of the most surprising evolutionary findings in neurobiology in, decades. For so long, the prevailing wisdom just assumed that IGLRs,
the workhorses for fast excitatory transmission in the brain,
the absolute workhorses, it was assumed they shared a common ancestor with the other major LGIC families, like, say, the nicotinic acetylcholine receptors.
Which makes intuitive sense.
It makes sense, but the evidence now just overwhelmingly supports a complete revision. That widely assumed relationship is, it's incorrect. IGLRs are something else entirely.
So if they aren't related to the other big ion channels, what are they? What is their lineage?
They're proposed to be mosaics, composed of several evolutionarily distinct modules, and the most ancient and compelling module, the one responsible for actually detecting the neurotransmitter glutamate, traces its lineage all the way back to the sensing machinery of gram-negative bacteria.
This is the famed Venus flytrap ancestry. We have to spend some real time here because the implication is just staggering. You're saying the very mechanism that allows you to learn and form memories started as a system for a bacterium to, find food.
That is the core realization.
Yeah.
Yes, the bacterial component in question is called the periplasmic binding protein, or PBP.
Periplasmic, so that's the space between the membranes in a bacterium?
Exactly. In gram-negative bacteria, it's the space between the inner and outer membranes. And these PBPs, they're essentially the cell's external scouting and scavenging force.
And what were their original functions? What did they do for the bacterium?
They're multi-talented, really. They bind this vast array of small molecules, sugars, ions, and crucially for our story, amino acids. Once a PBP binds a substrate, it does one of two things. Either it helps transport that nutrient into the cell, or it initiates chemotaxis,
Which is...
guiding the bacterium to swim toward the source of that nutrient. It's a guidance system.
And the Venus flytrap analogy, that helps us understand how they work, not just in bacteria, but in our own brains.
It really does. The 3D structure is very characteristic. It has two distinct globular lobes, and they're connected by these little hinge strands. When the PBP is open, it's just waiting. When the right molecule comes along and binds in the cleft between the lobes, the two lobes just snap shut around it.
Trapping it.
Trapping it. It's this precise, massive conformational change. And what's fascinating from that modular perspective is that this PBP gene module, it's been recognized in multiple, otherwise totally unrelated gene families across both prokaryotes and eukaryotes. You've just a highly successful reusable design.
Okay, let's nail down the specifics of the homology here, because this isn't just a vague structural similarities. There's direct sequence homology.
Oh, it's direct. Direct sequence and functional homology between these bacterial PBPs and the ligand-binding domain of our ICLRs.
So which bacterial PBPs gave rise to our key binding sites?
The sources outline this really specific two-part ancestry, and it's related to the size of the IGLOR subunits. First, you have the larger ones, the roughly 100 kilodalton IGLOR subunits. These are the most common ones you find in insects, amphibians, and mammals like us.
Okay, and what's their bacterial grandfather, so to speak?
The long N-terminus extension on these big 100 kilodalton subunits shows this really clear sequence and structural similarity to two bacterial proteins, the leucine, isoleucine, the lean-binding protein, or LIVBP,
LAVP,
and the leucine-binding protein, or LEUBP. These are the bacterial specialists for branched-chain amino acids.
So a protein for scavenging leucine somehow became a part of our main excitatory neurotransmitter receptor.
That's right. And that N-terminal domain is now thought to regulate the trafficking and assembly of the mature IGLOR complex. But its physical homology is just, it's undeniable.
Okay, so that's the first module, scavenging leucine. What about the core glutamate binding site itself, the part that actually does the work?
Right, that comes from the second and even more critical link. And this one is present in both the 100 kilod subunits and the smaller 50 kilod subunits, like the kainate binding proteins you find in lower vertebrates.
And this domain is homologous to a what?
To the lysine, arginine, ornithine binding protein, LAOBP, and the glutamine binding protein, or QBP.
Wait, hold on. So that's two different binding protein families, LAOBP and QBP? Are you saying they're fused together somehow to create the modern glutamate binding site?
Essentially, yes. In the IGLOR structure, these LAOBP and QBP-like components are found in two really strategic places. One is in the N-terminus extracellular domain, and the other, critically, is in the loop that connects transmembrane regions 3 and 4.
So these two pieces come together to form the pocket.
They form the bipartite glutamate binding pocket. It's a literal fusion of ancient bacterial scavengers, repurposed to create this new, high-speed excitatory sensor in our brains.
And the success of this modular design meant evolution just. It used it everywhere. It needed an external sensor, didn't it? How broadly do we actually see this PBP ancestry in other key mammalian drug targets?
Its pervasiveness is astonishing. I mean, it seems like whenever evolution needed a sensitive extracellular ligand sensor that could undergo a major conformational change, it just repurposed the PBP design.
So what are some other examples?
Well, the sources confirm this module is critical in, first, the metabotropic glutamate receptors, the MDLRs.
Which are GPCRs. That shows how one binding module can be coupled to two completely different kinds of channels.
Exactly. Then you have specific components of the GABA-B receptors, GABA-BR1A and BR1B.
Our main inhibitory system.
Main one. Then there's the calcium-sensing G protein-coupled receptors, the KSRs. Even the natriuretic peptide receptors, the AMP receptors, which regulate blood pressure and cardiac function.
So a piece of molecular machinery that evolved to pull nutrients out of, pond water. Now it regulates your heart rate and your ability to detect pheromones, given the similarities to KSRs and EMBLRs. The efficiency is just breathtaking.
It really speaks volumes about evolutionary constraint. If a mechanism works, you reuse it. This common PBP structural motif is what allows researchers today to build reliable homology models for all these diverse mammalian ligand-binding domains.
It links everything back?
It links everything from calcium homeostasis to inhibitory neurotransmission all the way back to bacterial nutrient sensing.
This structure must impose some really strong constraints on how the receptor actually works, though. I mean, how does that venous flytrap movement get translated into the physical opening of the ion channel?
That is the crucial mechanical step. So you have the PBP-like domain, the binding pocket sitting externally. Inserted between the two lobes of this flytrap are the three transmembrane segments, M1, M2, and M3, which are what form the channel pore.
So the insertion point is key.
The insertion occurs right near the interlobe crossovers in the bacterial PBP structure.
Ah, okay. So when the two lobes snap shut around a glutamate molecule, that structural force is directly pulling or pushing on the transmembrane helices below.
Exactly. The composite movement of the two lobes coming together is mechanically coupled to the movement of those membrane helices. And this conformational shift is what's necessary to widen the central pore and allow ions to flood through. Understanding the bacterial PBP model is what gave us the framework for understanding the activation mechanics of the entire iGlora channel complex.
So we've established the origin of the ligand binding site. Now we have to look at the keyhole, the ion channel pore itself. So is the ion conducting part also borrowed from somewhere else?
Yes, and this is what really demonstrates the true chimeric nature of the iGlora. Once evolution had perfected the PBP sensor, it needed to connect it to a reliable ion conduit.
So it went shopping for another part.
It did. And the channel-forming region of iGlorars, specifically the sequence between transmembrane segments 1 and 3, shows this surprising, deep evolutionary relationship with channel families that, on the surface, look totally unrelated functionally.
We're talking about electrical channels, right? Not ligand-gated ones.
We are. The sources detail these really striking structural and evolutionary similarities between the iGlorar pore and the pore lining segments of voltage-gated K+ channels.
Like the famous shaker K+ channel from fruit flies.
The very same. And also cyclic nucleotide-gated channels, or CNGCs. Now, this is totally counterintuitive, right? Shaker channels respond to changes in voltage. CNGCs respond to internal second messengers like CAMP. Neither of them responds to extracellular glutamate.
Yet the pore architecture is shared.
The pore is shared. The structural constraints of reliably conducting potassium ions led to a common design that just worked.
So what are the specific molecular fingerprints that link these disparate channels together? What's the smoking gun?
The sources point out 4 crucial conserved amino acid positions that are shared among all 3 families: K+ channels, CNGCs, and iGlors. This includes a highly conserved pair of adjacent aromatic residues that are just vital for channel function.
And there's more. Right. There's a specific motif.
Yes. Beyond that, iGlorars possess this specific critical sequence motif that mirrors features of the K+ channel pores.
Tell us about that conserved motif and what it does.
It's known as the QRN site, a sequence of glutamine, Q, arginine, R, and asparagine, N. You find it in subunits like the RAC-GLORAR and GLORAR2.
And it's centrally positioned in the pore.
Right in the middle, and it plays this huge functional role. In K+ channels, the pore architecture defines ion selectivity. In iGlorars, this motif is what determines internal blockade in a phenomenon called inward rectification.
Meaning it conducts current more easily into the cell than out of it.
Right, which is a necessary function for excitatory signaling. So this very specific mechanism of ion regulation is deeply conserved from the K+ channel family.
So even the subtle functional mechanics of how ions flow through the channel are borrowed from an ancient K+ channel blueprint that originally evolved to respond to electricity.
It is a remarkable example of nature mixing and matching. The extracellular phase is a bacterial nutrient scavenger. The transmembrane domain is an ancient K+ channel conduit. Put them together, and you get the iGlor, a brand new molecular machine for higher cognition.
Just incredible.
And we even see further conservation. There's a conserved aspartate residue that contributes to a negatively charged ring at the external mouth of the pore, which probably helps attract positive ions, just like in other channels. The evidence for these LGICs as chimeras, built from successful pre-existing components, is just overwhelming.
That sets a phenomenal stage for modular evolution. We've looked at systems built from repurposed scavengers and electrical conduits. Now let's transition to the other massive superfamily of drug targets, the G protein-coupled receptors, or GPCRs. The ancestry here moves from scavenging to sensing light, the 7-helix saga, all centered on rhodopsin.
Right, rhodopsin is the original ancient blueprint for what we call Class A GPCRs. It's defined by that signature architecture of 7 transmembrane helices, or 7TM.
And its primary role was sensing photons.
Exactly. And in doing so, it established the conformational switch mechanism that literally thousands of human receptors now use to detect Chemicals, hormones, neurotransmitters, odors, lipids. It is, arguably the most successful molecular scaffold in all of biology.
How far back does this lineage go, and how did evolution actually tune the structure to see different things?
Well, the origins of vertebrate visual pigments, they trace back deep into the deuterostome lineage. The ancestral C-opsin gene underwent repeated gene duplications and then subsequent mutations. And this diversification led to whole families like OPN3s, parietopsins, and eventually the ancestral vertebrate visual opsin.
Giving early organisms this wide spectrum of light they could sense.
Exactly. And the tuning process itself is beautiful proof of functional selection. It's precise molecular evolution. Opsins are tuned to absorb different wavelengths of light by making these really subtle changes to the environment around the bound light-sensing molecule, 11-cis-retinal.
So a few mutations can change the color you see.
A handful. For example, specific mutations at just 5 key sites in the LWS, or long wavelength sensitive opsins, can shift the absorption peak dramatically. The unbound 11-cis-retinal on its own absorbs UV light, around 380 nanometers. But when you covalently bind it inside the protein pocket and protonate it, the electronic cloud spreads out, and that peak shifts way into the visible range, allowing us to see color.
Let's focus on the activation mechanism, because this really is the template for almost every GPCR drug we use today. What is the molecular choreography when rhodopsin detects a photon of light?
Okay, so in the dark state, you have 11-cis-retinal covalently linked to the receptor. When a photon hits it, it triggers this near-instantaneous photosommerization. The 11-cis-retinal literally flips its configuration to all transretinal. That flip is a trigger.
And that structural change must completely destabilize the resting state of the protein.
It does. The change propagates rapidly through the whole helix bundle. What follows is what's called the meta-nia-meta-2 transition, and this happens in about a millisecond. The first major event is the deprotonation of the shift base bond holding the retinal in place.
And that's what causes the color change.
That's the spectral shift. It goes out of the visible range and back into the UV.
And the final signaling state, the one that actually recruits the G protein and starts the cascade.
That is the active state, known as R star, or meta-IIBH plus the attactor. To get to this state, two critical things have to happen on the cytoplasmic face of the receptor. First, there's a substantial outward movement of transmembrane helix 6, TM6.
By how much?
About 10 to 15 angstroms. You can think of TM6 as a lever that just swings out to expose the G protein binding site.
In the second step.
The second step is a proton is taken up from the intracellular fluid to neutralize a specific residue, glutamic acid 134. This neutralization, combined with the TM6 movement, forms the fully activated conformation that can then catalyze the exchange of GDP for GTP on the G protein transducin. That's what initiates the whole visual cascade.
That's a highly specific, repeatable activation model. So once that blueprint existed, the GPCR family just exploded. And it shows both massive divergence of function, but also these fascinating examples of convergence on ligands.
Absolutely. I mean, take the cannabinoid receptors, CB1 and CB2. They're classic examples of functional divergence. They're reusing that 7TM scaffold, but for chemical signaling instead of light.
And they respond to endocannabinoids, like an andamide.
Right, and their mechanism is strictly canonical. Ligand binds, G protein activates, and then you get desensitization via phosphorylation by GRKs and recruitment of beta-arrestin, the whole textbook mechanism. And CB1 in particular is one of the most highly conserved receptors among all vertebrates. You can find it in very basic invertebrates.
But the really curious evolutionary twist is convergence, isn't it? Where completely different branches of the GPCR family independently evolved to detect the same signaling molecule.
Yes, and the lysophosphatidic acid, or LPA receptors, are a prime example. LPA is a very common lipid signaling molecule. We have the established LPA receptors, LPA1, 2, and 3. But the sources highlight two others, GPR23, which is now reclassified as LPA4, and GPR92, which is now LPA5.
And phylogenetically, they aren't closely related to the original LPA receptors?
Not at all. They're only distantly related. And yet, both of them have evolved the capability to respond to and be activated by LPA.
So LPA was the successful, crucial signaling molecule in the environment, and multiple separate evolutionary lines within the GPCR superfamily just happened to stumble upon a way to detect it.
It reinforces the idea that there are, limited effective ways to communicate chemically, and evolution will repeatedly find the same successful solution. This Convergent evolution toward a pre-existing signaling molecule is just a powerful demonstration of natural selection at the molecular level.
Before we move on, what about the most basal use of GPCRs? Where do we see this 7-helix architecture acting as a basic chemosensor in lower life forms?
The diversity is just immense. You see, invertebrates like Drosophila, the fruit fly, they often rely on entirely different types of receptors for environmental sensing, like those non-GPCR variant iglorars, which shows there are alternative solutions to the same problem.
But the lineage of our own senses, that appears pretty early in the deuterostomes.
Does, like our sense of smell. Vertebrate-type olfactory receptor genes, the OR genes, which are the true precursors to our sense of smell, they've been identified in the cephalochordate Branchiostoma floridae,
A lancelet.
a simple lancelet. This basic organism marks the point where that 7TM architecture truly begins to specialize in detecting volatile external chemicals, which led to the massive expansion of the OR family in mammals.
And what about the surreal GPCRs and echinoderms like sea urchins and starfish? That name alone suggests a pretty unique evolutionary niche.
It does, doesn't it? Echinoderms have this unique repertoire of OR-like GPCRs, often called surreal GPCRs. And they're typically expressed in tissues that directly contact the ocean environment, the pedicellariae, the tentacles, the body wall.
So they're smelling the water.
In essence, yes. Their function seems to be highly specific environmental chemosensing. They are the ancient marine equivalent of a complex nose or a tongue, adapted to sense the subtle chemical changes in the water. It's yet another example of the 7TM fold being adapted for novel sensory modalities long before the complex regulatory roles we see in us.
Incredible. Okay, we've covered membrane receptors built from bacterial parts and light sensors. Now let's pivot entirely inward. Let's look at the receptors that govern intracellular signaling and gene transcription, the tyrosine kinases, or TKs, and the nuclear receptors, or NRs.
Right, so we're moving from the surface sensors to the master command centers inside the cell. And tyrosine kinases are absolutely fundamental. They initiate these phosphorylation signaling cascades that regulate, well, virtually every cellular process. Growth, death, everything.
And they're all united by sharing a highly conserved catalytic domain, right? The TK domain.
That's right, paired with varied non-catalytic domains that dictate their specific function.
So if they're so universal, how ancient is the sophisticated Peter signaling system?
Well, the complex tyrosine phosphorylation system, we can pinpoint its origin to pre-Metazoan unicellular organisms. We're talking about creatures like ichthyosporians and corallichytrium.
So these are close relatives to animals, but they're still single-celled.
Exactly. And they were already using this elaborate system of communicating by adding phosphate groups to tyrosine residues, a system that far preceded the evolution of complex organs or tissues.
And within that ancient system, the core cytoplasmic kinases, the CTKs, were already established. They show deep conservation.
Yes. The four main families of cytoplasmic tyrosine kinases, Ceriarchaea, Abel, Poinsk, and Tek, are remarkably shared across metazoans, choanoflagellates, and Filisterians. This suggests that the earliest diversification of the Holozoic clade already contained a repertoire of CTKs that looks surprisingly like what we rely on today for basic cell maintenance and signal processing.
They are the essential, deeply conserved machinery that never really needed much change.
They got it right early on.
But the receptor tyrosine kinases, the RTKs, the ones that span the membrane and sense external ligands, they show a very different evolutionary pattern. It's not conservation, it's explosive radiation.
That is the striking contrast. While the CTK complement was stable and conserved, the RTK repertoire showed these massive lineage-specific expansions.
Yeah.
Ichthyosporians and corallichytrium possess a much larger number of RTKs than CTKs.
And crucially, their external domains are different from ours.
Completely different. These pre-Metazone RTKs have unique architectures. They're membrane-standing, yes, but their external ligand-binding domains are not homologous to any known Metazone RTK family.
So the catalytic engine, the TK domain, was conserved and ancient, but different lineages independently attached all sorts of different external antennae to it.
Precisely. It means the idea of building a membrane-spanning receptor coupled to a TK domain was a successful architectural plan that was discovered repeatedly. The expansion and functional specialization of RTKs happened separately in many different lineages as they evolved to sense different signals, like growth factors and hormones.
Okay, now let's move to the nuclear receptors, the NRs. These are the receptors that regulate transcription, the ultimate control system. Here, the sources describe domain swapping as the key evolutionary mechanism.
Right. Nuclear receptors are defined by their ability to go into the nucleus and directly regulate DNA transcription. and they all share a common progeny. Structurally, they're defined by their highly conserved DNA-binding C domain and the more variable transcriptional or ligand-binding E domain.
So what does domain swapping actually look like in this context?
Well, phylogenetic analysis shows that the history of NRs is really complex. It involves extensive gene duplication, followed by the functional exchange of these conserved domains. Normally, you'd expect the phylogenetic tree for the C domain to mirror the tree for the E domain if the whole gene was just duplicated together.
But they diverge. They don't match up.
They don't, which is clear evidence of molecular shuffling. We see examples like the Drosophila nerps family, where different species acquired very different E domains, which allows them to bind new ligands or interact with different cofactors, all while keeping a similar DNA binding profile.
And the vitamin D and exosome receptors are another example.
Classic example. Their C and E domains actually belong to different phylogenetic classes, which indicates a definite swap sometime in their history.
So evolution figured out how to mix and match a regulatory element with a transcriptional element to rapidly create new gene switches.
Yes, and this mechanism allowed NRs to rapidly diversify their regulatory and ligand-binding roles. They became sensors for everything from steroids to vitamins, tuning metabolism and development all across the metazoans.
Let's use the NR2F family, which governs immunity and metabolism, as a high-detail example of how this ancient scaffold takes on critical modern roles, including in disease.
The NR2F family is a perfect illustration of this functional divergence. If you take NR2F2, for instance, while it has routine roles in regulating things like adipose tissue, the sources really emphasize its subversion in pathology. It acts as a major facilitator of prostate tumorigenesis.
How does it contribute to cancer? What's the mechanism?
It achieves this by actively inhibiting the TGF-beta-induced growth barrier. TGF-beta signaling is crucial. It usually acts as a safeguard. It can trigger growth arrest or apoptosis, especially in early-stage tumors.
And that barrier relies on SMAP.
It relies on SMAT4-dependent transcription, and NR2F2 functions as a transcriptional repressor. It essentially overrides the TGF-beta signal by blocking SMAT4 transcription.
So an ancient core regulator gets hijacked to turn off the body's natural brakes on cancer cell growth.
Precisely. Its overexpression or hyperactivation is strongly correlated with tumor recurrence and disease progression. It shows how an ancestral regulator, meant for maintenance, can be co-opted into a driver of malignancy just by inhibiting a key tumor suppressor pathway.
And NR2F6, its family member that fine-tunes the adaptive immune response, also uses a really complex molecular switch.
Yes, NR2F6 is crucial for balancing the immune response. It can act as either a transcriptional repressor or an activator, regulating the differentiation and function of multiple T-cell subsets. For instance, in T-cells, it generally acts to suppress the machinery that produces inflammatory cytokines.
So if you get rid of it, the immune system goes into overdrive.
That's what you see. If you deplete NR2F6 in mice, they show increased accumulation of follicular T helper cells and germinal center B cells. It's an overexuberant immune response.
But how is it regulated during a real immune challenge like an infection? The T cell has to be able to turn on.
It does. The brakes have to be released. And this is achieved through regulatory phosphorylation. When a T cell is activated, a specific protein kinase, PKC theta, targets and phosphorylates a specific residue, serine 83, which is located right within the DNA binding domain, the DBD, of NR2F6.
Phosphorylating the DBD itself sounds like a brilliant way to immediately turn off its function.
It is. This phosphorylation negates NR2F6's ability to bind to the DNA loci of cytokine genes. With the NR2F6 brakes released, other key transcription factors like NFAT and AP1 are free to bind to the DNA for powerful cytokines like IL-2 and interferon gamma.
And you get rapid transcription and secretion.
Exactly. It's this really complex feedback loop that shows how an ancient nuclear receptor has evolved to provide this precise, on-demand suppression and activation that's necessary for modern adaptive immunity.
We've traced the evolution of channels, sensors, transcriptional governors. Let's conclude our deep dive with a system that is central to both pain and addiction, the opioid network.
The mu-opioid receptor, or OR system, is an ancient and crucial part of our survival toolkit. Its evolutionary function is deeply centered on maintaining endocrine homeostasis and promoting adaptations during challenging environments, pain, trauma, severe stress, fasting.
And these functions, linking stress and pain, are highly conserved, right? You find them in invertebrates, too.
You do. And the endogenous ligands, our body's own painkillers, are linked to other major stress hormones, which really reinforces that deep evolutionary connection.
Right, the precursor protein, POMC.
Exactly. The common precursor protein for beta-endorphin, which is the endogenous ligand for MRR, is pro-opiomelanocortin, or POMC, and POMC is also the precursor for ACTH, adrenocorticotropic hormone.
So the body links the stress response directly to the pain response at the molecular level.
Right. When the body activates the POMC pathway because of stress, it simultaneously generates molecules to regulate the stress axis, ACTH, and molecules to suppress pain, beta-endorphin. The MOR system is just deeply embedded in these maintenance programs that help us prioritize resources and dampen suffering during a crisis.
Now, the sources emphasize that the complexity of opioid signaling goes far beyond just having 3 different receptor types, mu, kappa, and delta. The true regulatory complexity is in how the gene itself is handled.
That's the molecular trick. All the complexity of the mu-opioid receptor often arises from the alternative splicing of a single gene, OPRM1.
So different combinations of exons are stitched together.
Exactly, and this generates a whole plethora of structurally and functionally distinct mu-opioid receptor variants.
What kind of structural diversity are we talking about here?
Oh, this splicing yields versions ranging from the canonical full 7-transmembrane helix variants all the way down to truncated forms. You get single transmembrane variants, variants with entirely different C-terminal tails, and critically, these variants often have distinct biochemical properties.
So they might couple differently to G proteins or desensitize at different rates?
Exactly, or even signal via different internal pathways. They effectively constitute an entire network of receivers, all operating from a single genetic instruction set.
And what mechanism does the cell use to fine-tune the exact ratio of all these different variants?
One key mechanism the sources highlight is nonsense-mediated mRNA decay, or NMD. This is a surveillance pathway in the cell, and its primary job is to recognize and degrade mRNAs that contain premature termination codons.
To prevent the production of harmful truncated proteins.
It's precisely. And alternative splicing often generates transcripts that contain these premature stop codons. In the mouse OPRM1 gene, researchers have identified 11 different splice variants that are potentially regulated by NMD.
So when NMD degrades those transcripts, it explains why some of the variants have very low expression levels in the brain.
Exactly. The cell is actively filtering the output of the OPRM1 gene, making sure only the desired variants, or at least only some of the potential variants, reach high expression levels.
So the final function, whether it's pain relief or addictive potential, is the outcome of the specific combinational expression of this one gene, regulated by mechanisms like NMD.
That's it. The local environment, a specific brain region, a specific tissue, determines which splicing factors are active and which NMD targets are degraded. And the final cocktail of MOR variants is what determines the ultimate characteristics of that receptor network.
Including its sensitivity to opioids and its propensity to trigger addiction.
Right, and also the long-term side effects.
Which brings us back to that drug-target paradox. I mean, we were effectively treating pain with opium for millennia, preceding any molecular understanding of this intricate network.
Indeed. We had potent drugs long before we understood that a single gene could produce dozens of receiver variants regulated by these ancient surveillance mechanisms. This history really underscores why studying deep evolution is so critical for pharmacology.
If we understand the ancient function, which was coping with stress and famine, we can better understand the modern pathologies.
And those modern pathologies can be really far-reaching, like the effects on bone metabolism. The persistence of opioid engagement alters endocrine homeostasis beyond just the pain axis. We're still uncovering the full spectrum of consequences. It all comes back to that combinatorial expression of ancient, highly adaptable receptor components.
Okay, let's try and synthesize this incredible journey. We started with the modular nature of life and found bacterial nutrient sensors at the heart of our brain. We moved through light-sensing opsins, and we ended up with transcriptional regulators built through domain swapping.
The overarching synthesis is, I think, very clear. The most crucial drug targets in your body, the ones mediating brain function, immunity, endocrine control, are evolutionary mosaics. They're patchworks.
Aglagars and mglars use that venous flytrap PBP module from bacteria.
The entire GPCR family is built upon the structural foundation of light-sensing rhodopsin. And intracellular signaling maestros like TKs and NRs show how ancient gene domains expanded and swapped capabilities to tune cellular response over billions of years.
So the central takeaway for you, the learner, is that the molecular parts we target with drugs today evolved hundreds of millions or even billions of years ago for tasks as simple as scavenging amino acids or responding to a photon of light.
It's the ultimate recycling program. Evolution didn't reinvent the wheel, it just, welded an ancient electrical conduit to a bacterial sensor, or it found a way to link an ancient signaling engine to a brand new external antenna. This modularity is why evolution could create such complexity so quickly.
And that modular dynamic nature brings us back to our starting point with rhodopsin, and it leaves us with a final provocative thought for you to explore. We talk in detail about the precise choreography of rhodopsin activation, that transition from meta-I to the truly active state, R star.
Right. And structural biology has confirmed the structure of the dark resting state very, very well. But capturing that transient, light-activated R-star state is incredibly difficult. So what's often studied in the lab is the decay product of meta-2, which is the apoprotein-free opsin, often called OPS. And this OPS state, while it's mostly inactive, it still shows some residual signaling activity, which we call OPS-star.
And the sources raise a critical structural paradox about those two active states.
The central uncertainty is whether the light-induced R-star state, the natural true signaling state, is molecularly identical to the OPS-star state that's derived from decay or stabilized with synthetic ligands. There's actually strong evidence to suggest they might genuinely exhibit different activities, different kinetics, and potentially different downstream signaling biases.
We still don't have a definitive picture of the true light-induced R-star state.
Not at high resolution, we don't.
So, the thought to take away is this. When modern chemists synthesize a drug designed to hit one of these ancient, highly dynamic GPCRs, say, a drug for pain or blood pressure, are they synthesizing a molecule that locks the receptor into its true intended natural signaling state, the R-star state? Or are they stabilizing a slightly off-kilter decay product, the OP-star state?
The implication is immense.
It is. Even at the center of the world's most successful drug targets, there is still fundamental ancient molecular biology yet to be uncovered, which means better, more targeted drugs are still waiting to be designed.
A fascinating challenge for the future.
Indeed. We'll see you next time for another Depth Dive.